So can I start? Dr. 𝐀𝐝𝐻 Prasad. So I feel very elated to talk about Sir, being our resource person today. Dr. 𝐀𝐝𝐻 Prasad, sir, who is a respected authority on scientific officer, grade 4, Liquid Dielectrics Laboratory, Central Power Research Institute, Regional Testing Laboratory, Noida. Sir has pursued his PhD on polymer sciences technology in 2010 from SK University, Anantapur. His research focuses on synthesis and characterization of polymeric matrices for controlled drug release and evaporation applications. He is a recipient of meritorious fellowships UGC, New Delhi and Brain, Korea, with 21 prestigious fellowships at South Korea. Sir has published a book titled on vegetable oil based on biolubricants and transformer fluids in Springer Nature, Singapore. Sir holds very reputed patent on green insulating fluids for transform applications, a non-edible vegetable oil. To add to his credits, he has published over 32 international journals and national journals. He has attended national conferences of about 11 and 9 international conferences. Sir is very passionate about solid and liquid insulators for transform applications, polymer blend and composites, and polymeric membranes for fuel cells and evaporation applications. Sir has also worked as co-investigator and investigator in IHRT projects with innovative titles and with high outlays. Sir's research experience has played an integral part with several prestigious positions like Scientific Officer Grade 2, 3, and 4 at Bangalore and Noida. He has also received a postdoctoral researcher and research professor in Changwon National University, South Korea. Presently, Sir is discharging his duty as a scientific grade 4 at Central Power Research Institute, Regional Testing Lab, Noida. Sir, we are very fortunate to have you with us today as the resource person, and we are very thankful for you for accepting our invitation and being the resource person. With the passion of innovation and excellence, Dr. 𝐀𝐝𝐻 Prasad has established himself as a leading person in the scientific field. So welcome, sir, and I request you to please take over the session.
Thank you, madam, for your introduction.
Welcome, sir.
So I'm, as madam brief about me, I'm currently working as a scientific officer in Central Power Research Institute, Noida. Prior to joining here, I worked as a postdoctoral for 2 years in South Korea. So in 2013, I have joined in Central Power Research Institute. Till 2018, I have worked in Bangalore. And from 2018, June onwards, I am working in Noida. So before going to our actual presentation, I would like to brief about CPRI, which will be very useful for even faculty and students who are working in the areas of electrical, mechanical, and chemical fields. Because actually our institute is mainly working on this engineering background, so it will be helpful for you. So just I would like to brief about the institute. So CPRI is an autonomous body under Ministry of Power, which is established in the year 1960. And CPRI is having various laboratories across India, such as Bangalore, Bhopal, Hyderabad, Noida, Kolkata, Guwahati, and Nagpur. And 2 more branches in Guwahati and Raipur and Nasik is coming in one or couple of years. And actually what we are doing in our CPRI is, we are doing research and development activities pertaining to power sector applications, testing and servicing, providing services to the state electricity boards, manufacturers of power equipments, and electrical, mechanical, power sector. And we are also doing collaboration related to power related instruments. And we are doing the consultancy work regarding the transformers, various applications of elements. we are doing consultancy in various areas from power laboratory, material design, and other consultancy also we are doing. So who are facing the issues in the ground level cases equipments pertaining to power sector. And we are also certifying the product certificate for the we are testing laboratories across India or NABL accredited laboratories related to our faculty and other students also. This careers ranging from which having done their ITI, ranging from ITI to PhD careers. So we are also doing R&D collaboration with various institutes like IIT Mandi, IISc Bangalore, and other research institutes. So if anybody interested, even in the teaching people also, they can have collaboration with the CPRI expert, and then we can giving funding also own people also can or collaborate with CPRI officials, and we are also VTU is registering and who are registered under VTU, they will be providing fellowships and doing their work completely in CPRI. So, and we are also providing MTech projects for various areas like mechanical, chemical, electrical, and other material related also. And we are mainly working electrical, chemical, materials, technology, and mechanical related. Electrical from LED bulbs to cable insulator, renewable energy, all we are doing testing as well as consultancy and R&D activities. And related chemicals, materials pertaining to power stations and liquid dielectrics also taking care of both R&D and testing. And regarding to materials also, we are doing a lot of testing. Polymers also doing testing. And simulated, please, for further details, you can visit our website on cpi.res. For various, you can this MTech announced during the month of January every year. Are doing just they are doing something. We have to finish the project like that. They are doing, but many people may be doing like that. But some people be doing. So I advise you people in order to get a fellowship projects so that we get best result over.
Now it is changed.
No. Actually, it is in normal mode. In presentation mode again, it is. No, sir.
No, sir.
Changing, no?
Sir, it's
No, sir.
normal mode,
So I
sir.
continue with normal mode. I will continue.
Yeah. Manually you can change.
Sir, first of all, I would like to thank to the management for the program to enhance the skills of the faculty, which helps them
Sir? Sir, sorry for the disturbance, sir. Your voice is not audible, sir.
this type of program good for both.
Sir?
Biodecompatibility and addition polymer for drug delivery application, novel drug delivery systems, mechanism of drug delivery system, targeted drug delivery system, and mucoadhesive drug delivery system. First of all, what is a polymer? polymer is a combination of many monomers that combined chemically to form a macromolecules. So this is actually this word has been derived from French word poly means many, mers means parts. Together is chemically combined together to form a polymer. Due to their versatile properties like lightweight, there other superior properties. These are polymers are using wide range of applications like food packaging, transportation, plastic piping, clothing like nylon, firing industries, and electrical industries and electronic industries. Circuit of polymers. So there is area where polymers involved. So everywhere there is a usage of from the day we get up from the bed, we have to start with the polymers. People think that plastics only are polymer problem to the environment. So if you go too deep into the subject, so a lot of applications will be there, like defense applications, aerospace applications, space applications, and a lot of other applications like biomedical applications also. So this is very important subject to know each and everyone nowadays as per the current scenario. So coming to the medical industry, polymers are widely used in various applications of medical industry, like drug delivery carriers as a membranes for delivering the drugs, in diagnostic devices like scanning electron microscope, vascular sensor for identifying the problem in the body, implantation like knee replacement and other scaffolds. We are using wide range of applications in polymer. Using these polymers, we are day by day it is the technology is increasing in medical industry. Because of this versatile polymers, can any change their structure properties by modifying these polymers. So what is the role of polymer matrices in modern medicine? First of all, what is a polymer matrices? Polymer matrix is a continuous phase which is combines of other drug or enzymes to the particular site. So it is a plays a key role in modern medicine, liver system, tissue engineering, and medical devices. And due to their versatility of properties, due to their biocompatibility in nature and ability to engineering to specific applications. So among these things, sustainability dosing frequency, it is used for dosing to reduce the dosing frequency and targeted drug delivery. So these are biodegradable hormones are and biodegradable.
Sir,
Stitches, they have to remove after 1 week or 10 days, they will remove that stitches. But nowadays, these are easily degradable and hence it will automatically removed off. So it is also used in a personalized drug delivery and in tissue engineering and regenerative medicines. These scaffolds for tissue regeneration, cell encapsulation, and biodegradable scaffolds also, these polymer matrices will be using. And this also used in wound healing and drug-loaded wound healing devices, which use in dressing and release the drug and improves the growth factors in the form of hydrogels and films. And as a site for control release at the site of where exactly the injury is happened, it will deliver the drug and improves the effectiveness. So it is also used in various medical devices like implants and prosthetics. Like suppose if anybody is having some injury or due to accident or by birth, if anybody is not having hand or leg, they will be replaced with prosthetics and implants also. So these are all used in control release coatings also for scanning electron microscope and other medical devices also. So these are also used in diagnostic applications where microparticles are using for imaging purpose. These are also used as a biosensor for detecting of various diseases like diabetes or cancer cells, in which normal testing methods we cannot able to identify these diseases. This is also used in vaccines and immunotherapies and vaccine delivery formulations like polymers as a immunostimulating agents and receipt as a specific receptors. These are also used as adjuvants, which enhances the effectiveness of the medical treatment. It is also used in non-injectable routes of drug administration like oral and nasal drug delivery, inhalable drug delivery systems in case of COPD patients. And the key characterization of polymer matrices is polymer-based structure. The matrix is primarily composed of synthetic or natural polymers such as polylactic acid, polylactic-glycolic acid, chitosan, or collagen. And the encapsulation or dispersion. The polymer matrix can be able to encapsulate the drugs, growth factors, cells, or other therapeutic agents, providing a controlled release or protection from degradation due to their swelling in nature and water solubility of the polymers. This also gives control drug release by degradation in the bio system by degrading controlled degradation process. So many polymer matrices are designed to gradually degrade in the body and release the encapsulated substances to the targeted site in controlled manner to degrade due to the they degrade due to the biological system present in the body. Coming to the types of polymers, these are classified as three types. One is natural polymers, synthetic polymers, and hybrid polymers. Natural polymers, these types are distinct characteristics suitable for various drug delivery applications. Each individual polymer has their own cause and concern. So natural polymers for drug delivery, mostly we are using chitosan, which is a bio-based material from cram and other seafood products, which is it will be extracted from the hard core of the seaweeds. So it is biocompatible and biodegradable. Because of its adhesive nature, it can be used as a mucoadhesive drug delivery systems also. It is widely used in drug delivery like oral, nasal, and topical applications also. It will enhance the drug absorption in the gastrointestinal tract due to their mucoadhesive in nature. And sodium alginate is one of the important polymer to form gels in the presence of divalent cations, example calcium. So actually in my PhD project, I have done based on these alginates only. So I have used this alginate polymer and grafted with other polymers and improved the release profiles. So these are used for controlled drug release like oral injection, tablets or injectable formulations also. Often these combined with other substances to form micro and nanoparticles also for improving the encapsulation efficiency of the drugs. Gelatin is one of the used particularly in the formulation of capsules, biocompatible drug delivery systems, and it is modified. You can also modify easily for controlled drug release also. And one more natural important polymer is hyaluronic acid. This is excellent biocompatible nature with our biosystem, and it is having good hydration properties. It is also used in injectable drug delivery systems, especially for targeting specific tissues such as joints and eyes. Starch and other its derivatives can be chemically modified, and as per the requirement, we can modify the structure and can be used as a controlled drug delivery system. Even it can be used as a a tablet formulation in drug delivery applications. The advantage of natural polymers is it is having biocompatibility. We can well tolerated by the body. If you give that formulations related to natural polymers, then body can resist because of its biocompatibility in nature and due to their biodegradability. These products will be degraded without any releasing any toxic effects, non-toxic byproducts, and reduce the risk of accumulation in the body. And these are the key benefits of these natural polymers. It reduces the risk of long-term toxicity or accumulation. When we give this type of drug delivery system to the body, it will not give, even if it is stay in the body for long time, it will not give any toxicity. And it will not be accumulated in the body. It can come through interact favorably with biological tissues due to their natural origin, because these are the discovered from the natural origin based material. This will be having good interaction with biological tissues and hence the therapeutic, because it will hold the tissues firmly and there it will release the drug. So it will be effectiveness will be more. So disadvantage of natural polymers is it is not widely available. So it is cost will be more. So it is one more other drawback with polymer, this natural polymers is batch to batch, because we are not, if you are synthesizing in a laboratory, we can maintain the same properties by choosing the combination, same compositions. But whereas in the case of natural polymers, it is not possible, because batch to batch it may vary, and depending upon the source material also it is varying. So this result may leads to the non-reproducibility of the result. So limited control over, you cannot modify the structure is, you cannot modify, because like molecular weight and drug release profiles, we cannot improve individually these polymers, and less mechanical strength and degradation rates can be harder to manipulate. If you want to degradation rate, vary the degradation rates, it is slightly difficult compared to synthetic polymers. It is potentially risk to immune reaction or allergic. It is not poisonous, but sometimes it may leads to allergic properties like proteins, gelatins, or albumins. And coming to the synthetic polymers in the area of drug delivery application, it can be designed with precise control. Whatever target, if you want to prepare which part you want to target that delivery, according to your requirement, you can modify these polymers. Whereas in natural, this facility is not possible. So we can control the molecular structure of the polymers. Their properties can be improved, and behavior at different conditions can be altered. So they offer more consistency and flexibility result than natural polymers and their physical and chemical properties. Each individuals have their own benefits. Some of the natural polymers have benefits, whereas some synthetic polymers have also some disadvantages. So combination of these two, we can improve the properties of the drug delivery system. So coming to the examples for synthetic polymers, we can most widely used polymer is polylactic 𝐀𝐜𝐴 acid. This is widely used. This is also biodegradable synthetic polymer that degrades into lactic acid and glycolic acid. These are naturally occurring in the body, so hence it is not toxic. So it is so this is the important polymer which we are using in current drug delivery devices. So polylactic glycolic acid is used in a variety of drug delivery systems, including injectable microparticles and nanoparticles, for the drug release to be sustained release for long time to be released. And one more polymer is polylactic acid and polyglycolic acid as a individual polymer. Whereas this one is copolymer, whereas this is individual polymers. These are biodegradable and commonly used in drug delivery system, and is oftenly used for long-term drug release. Suppose if you want to use the release the drug for long term, this type of polymers we can select. Whereas PG polyglycolic acid can be used for short-term applications. And this is one of the important polymer, polyethylene glycol. This is a non-toxic polymer and easily soluble in water. And it is can be used, modified the surface of the nanoparticles or liposomes to improve the stability and bioavailability and reduce the immunogenicity properties. So PEGylation is a process of modification of biological molecules by covalent conjugation with polyethylene. Is a common method to improve the pharmacokinetics of the polyethylene glycol. It is a quantitative analysis of the process of drug absorption, distribution, and elimination that determine the time course of drug action in response to the administered drug dosage. And one more important polymer, polycaprolactone. It is also a biodegradable and biocompatible synthetic polymer, which are widely used in controlled drug release device systems, particularly for sustained release of hydrophobic drugs. Where we are using hydrophobic drugs, this is the important polymer we can use. So one more, polymethyl methacrylate is a synthetic polymer which is used in ophthalmic applications for drug delivery, including intraocular implants and contact lenses also. And the advantages of polymer matrix is design flexibility. Synthetic polymers can easily design as per our requirement for specific properties such as degradation rates. Also, we can alter molecular weight. We can change. Suppose if the polymer having higher molecular weight, the drug release will be prolonged time. Suppose if it is low molecular, immediately it will release the drug. So, as per our requirement, the molecular weight of the polymers can be altered in case of synthetic polymers, and surface characteristics also can be improved as per our requirement and where we are actually using these drug delivery systems. And reproducibility of these polymers will be more compared to natural polymers. And his well-defined structure. This is our well-defined structure will be there. Whereas in natural polymers, the structure will be complex and hence we cannot able to modify their structures as per our requirement, leading to consistency performance of synthetic polymers. It is having wide range of properties like including solubility, mechanical strength, and degradation, and it can be precisely controlled to the suit of the drug delivery as per our requirement. And these properties can be controlled due to their by altering their molecular weight, degradation rate, and mechanical strength also can be controlled. The versatility, these polymers are versatile for designing systems with specific drug release profiles. If you want to a specific drug release profiles, either it is sustained release or it is prolonged release, we can use by changing these structures. And the cost is typically lower compared to natural esters and easier to scale up production. And here also some challenges is there with regard to synthetic polymers. Some of the synthetic polymers may not degrade easily and are not safely as that of natural polymers. And the degradation products may be toxic and leads to side effects. So this immunoresponse for few synthetic polymers may provoke immunoresponses, particularly in the case of prolonged implantation, or if you put in this type of synthetic polymer for long time implantation, it may lead to response will be less. Toxicity, these potentially toxic or adverse affection reactions may be possible due to the degradation of the synthetic polymers. And the compatibility is less compared to natural polymers. In some cases, compared to natural polymers, these are less biocompatible. So, in the environmental concern also, because these are non-renewable sources and are chemically processed involved in the synthetics, these are environmental concern is there. So, we have to take necessary approvals while processing these things. This is one of the concern of the synthetic polymers. So, based on this synthetic and natural advantage and disadvantage, the one more classification has been derived, that is hybrid polymer. This hybrid polymers comprises of both natural and synthetic polymer. Depending upon the requirement, we need which type of profiles. So accordingly, we can modify blending or copolymerization of these polymers, synthetic and natural combination. Suppose if you are having that mechanical strength is required, so higher composition of synthetic esters, synthetic polymers will be added, and biocompatibles also required means we will add natural polymers. And the nanoparticles and microparticles of this combination of this hybrid polymers can be also made. And the hydrogels also can be used mainly polyvinyl alcohol and agarose are one of the famous hydrogels combination used for sustained release and bioactive drug release purpose. And it has also helped in wound healing and tissue engineering applications. These advantages of hybrid polymers will give the combination of synthetic and natural polymers, and it balance the properties. Like, and a hybrid system can achieve better performance than each type of its own, because one synthetic polymer may be having some concern, and natural polymer will be having some advantages. So we can combine and get desired properties. So you can also expect improved drug delivery systems by using these hybrid polymers, such as drug loading capacity we can increase, release profile also as per our requirement, and targeting ability also will be improved, and the stability of the polymers with environment present in the body can be more improved. And these are the versatile, and hybrid can be used in a wide range of drug delivery application, from injectable systems to implantable devices. And the challenges of hybrid polymers it's a complex fabrication, because the structure of natural and synthetic polymers are entirely different. So in order to have the fabrication hybrid polymer, careful design and optimization is required to ensure the polymers are compatible and components and perform as required. So regulatory approval also is required, because combination of natural and because entirely different properties having synthetic and natural polymers. So regulatory approvals, it is very complicated, as both need to be thoroughly tested for safety and efficacy related issues. These are the properties I have already explained, and it is mentioned in the tabular column. For natural polymer, biocompatibility is more, whereas synthetic polymers, it can vary by changing its structure with and blending with other polymers. And hybrid polymers is high biocompatible when we choose the both natural and synthetic polymers of biocompatibility based polymers. And biodegradability, as the natural polymers are derived from natural sources, it is automatically naturally biodegradable. Whereas in case of synthetic polymers, may degrade slowly compared to synthetic ester, natural esters, and are more variable also. These hybrid polymers can be tailored as per our requirement regarding the biodegradability, and control over properties is limited for natural polymers, and precise control over molecular structure and the properties of synthetic polymers. And whereas in case of hybrid polymers, properties can be controlled, can be high. It is possible by changing the compositions of each individual polymers. Regarding the cost, I explained already. Examples for natural polymers is chitosan, alginate, gelatin, whereas for synthetic polymer, polylactic glycolic acid, PEG, polycaprolactam, polymethyl methacrylate, and hybrid polymer, PEG, PLGA, chitosan, PLGA, polylactic glycolic acid, gelatin, polyethylene glycol. And application is regarding the natural polymers is concerned, we can use it in wound healing, oral drug delivery, and vaccines. And whereas synthetic polymers can be used in sustained release and implantation, gene delivery, and have low risk of allergic properties and potential toxicity due to degradation products. And targeted drug delivery controlled release systems can be used hybrid polymers and also in tissue engineering. And the biodegradable polymers now we can go to biodegradable. What are the biodegradable polymers? These will this is with the aim of giving idea to the formulations. I am chosen this subject and importance of polymers in the biomedical application I am telling. So whoever is working in this area, it will be helpful for them, or in the future if they want to go towards formulation side, it can be useful. So I'm each point by point and which polymer, what is the role of these polymers in the biodegradable devices, I am explaining. So polysaccharides already told us, chitogen and alginate are naturally available polymers. Proteins and peptides is like albumin, gelatin, and collagen are natural polymers that can be used for controlled drug delivery application. They are also degraded in the presence of enzymatic process also. Because of the enzymes present in the body, it will undergo degradation and provide biocompatibles and biodegradable matrices. Polyurethanes and polycarbonates. Some polyurethanes and polycarbonates are also biodegradable, which can be used in drug delivery, particularly for control release over longer period. You can use this polyurethane and polycarbonates. And regarding biodegradable polymers, polyesters like polylactic acid, polycaprolactone can be used. In polyanhydrides, polymers such as polysebacic acid and polyesters can be used as a which can be degraded by hydrolysis of the anhydride bonds, and it can be used in various control release systems. These are typically degrade faster than polyesters and are often used in applications requiring where it is required rapid release of drug is required. This type of polymers can be used. And regarding the compatibility and biodegradability polymer matrices, drug delivery, if you come, ability of a polymer to be in contact with the body without eliciting any adverse reactions is called biocompatibility. Whenever the polymer is having biocompatibility, that means it can hold the biological specimen inside the body and it will not any adverse effects with body. That is biocompatible. Both are compatible. This polymer and the biological system present in the body. And key considering while considering key consideration when you are selecting biocompatibility is you have to check that non-toxicity, non-immunogenicity, cellular compatibility, lower inflammatory response. Also, these points we have to take care while selecting these polymers for drug delivery applications. Whatever the product, the biocompatible polymer you are selecting, it should be not toxic by degrading the when degrades in the body and forming that bioproducts should not be toxic and should not be metabolized or degraded in the body. So it should not have trigger immunogen response such as inflammation, allergic reactions also should not be there when selecting such type of polymers. We have to select for these systems, and it should be compatible with your cellular cells adhesion and proliferation, particularly for applications in tissue engineering or wound healing, where cellular interaction is essential. So it should be low flame polymers which are selecting should be low inflammatory response. And optimal polymers should be well tolerated by the body, allowing localized or systematic drug release without causing any chronic inflammatory problems. Examples of biocompatible polymers is regarding that biocompatible, both natural polymers and synthetic polymers are there, which is biocompatible with the system. These are almost natural polymers I have explained what the same polymers are widely using in this biocompatible as a biocompatible and biodegradable polymers. The biodegradability of polymer matrices ability is a breakdown naturally into harmless byproducts is called biodegradability. So in the presence of organic molecules, it should not break easily. It should break easily. Degradation, the drug will be released. So after releasing completion of the drug, should not affect the when it is after releasing the drug, it should not give any toxic effects in the products like water, carbon, and small organic also will be formed. So which will be easily throughout to the system. And we have to consider this enzymatic effects, pH, and other physical factors also we have to consider by selecting these biodegradable polymers. Eliminating long-term accumulation, we can issue over time, leading to chronic inflammatory fibrosis or toxic building enzymes that the material breaks down and its removal from the body after drug delivery is complete and reduces the risk of long-term toxicity. These are to be engineered to degrade at a specific rate, enabling the control result. So whichever we are using the polymer for drug delivery application, it should be degrade slowly. It should not increase. It should not degrade fast, or it should not degrade delay. So according to that, you have to can make the formulations. So it should be compatible with body metabolisms. So accordingly, these polymers we have to select. Naturally occurring metabolites that are safe for the body to proceed and eliminate. These byproducts are typically eliminated by urine and breathing. While selecting the biocompatibility polymers, we have to take that into consideration, these 3 points: degradation rate, metabolites, and environmental sensitivity. So degradation should be match and the desired drug delivery application. A fast degradation polymer might release the drug too quickly, and if it is slow degrading polymer, it may take long time to release the drug and lead to delayed therapeutic effects. It will not affect much on the to cure the patient. So the degradation products should be non-toxic with the biocompatible polymer. Whatever the products derived using the degradation, it should be compatible with the body. And these are the ideally these metabolites should be naturally or easily extracted by the body. For example, lactic acid and glycolic acid. As I explained earlier, when we use glycolic acid and lactic acid in copolymer, it will undergoes degradation and forms lactic acid and glycolic acid, which is not causing any harm to the body. So whatever the polymers we have to choose for this type of things, it should be having environmental sensitivity also. Should not affect any environmental conditions in the body, such as temperature, pH, or any other enzymatic item, and it should not influence the release profile of the drug. Examples of biodegradable polymers are polylactic acid, gelatin. So biodegradation mechanism is of 2 types. One is hydrolytic degradation and enzymatic degradation. Commonly, synthetic polymers like polylactic glycolic acid, where water molecules slowly break the polymer, change into smaller and soluble components. When interacting with this polylactic glycolic acid with water, it forms small molecules which are soluble in the system itself. So it will degrade accordingly. So enzymatic degradation is polymers like chitose and hyaluronic acid degrades to the enzymes present in the body. It undergoes reaction with the polymers undergoes reaction with the enzymes present in the body and can degrade. And this can be faster or more controlled in a certain environment. Accordingly, if you want, we can choose the system accordingly. The advantage of biocompatibility is reduced toxicity, and it can be tailored as per the drug delivery system, as per our requirement for like sustained release, or suppose if you want to long-term release of the drug, then and targeted, suppose specific target we have to reach the drug, then in that cases we have to tailor a drug delivery system accordingly. You can easily tailored. And patient safety also, because this will not eliminate any any toxic item, non-products, byproducts. So it will be safe for the patient. And reduce need for surgical removal. As I told earlier, in earlier days, they used to remove, after surgery, they used to ask to come for removing of the sutures. But nowadays, this type of polymers we are using, hence it is not required to remove. So automatically it will biodegrade when it contact with the body itself due to the presence of that enzymes, and it will undergo degradation. And there is no required to remove by surgical after releasing the drug. And the challenges and the consideration while choosing this biocompatible and biodegradable polymers is potential for incomplete degradation. Some of the polymers may not fully degrade, hence it may be lie down in the body, and which will affect the body system, which leads to the inflammation and irritation problems. While using this biocompatible polymers, some of the biodegradable polymers might still trigger to immunoresponse and cause toxicity during the degradation process. So you have to take care while choosing these systems. For example, if you are the degradation products affect the surrounding tissues or organs in unexpected ways, it cannot be suggested to make such formulations. And the stability of polymer or drug complexes will be taken into consideration while making this choosing this type of polymer. So this may affect sometimes interact with the polymer matrix, affecting its stability and bioavailability also, and further leads to degradation profiles also. So therefore, the interaction between the polymer and drug need to be thoroughly studied, whether it is compatible with the polymer, whether the drug selected is compatible with the polymer, or we have to ensure the efficacy and safety of the system also. And while selecting these polymers, how it is possible, whether it is possible to make this type of products which are without affecting the systems in the body, then we have to take care of these challenges also will be there while choosing this type of polymers. And because of the degradation rate can be complex and may require advanced techniques for optimizing the process parameter. These various applications of polymers in biomolecules can be considered as biodegradable polymers. Examples are like explained. These polymers can be used in tissue engineering, like scaffolds for tissue regeneration, and drug delivery applications like control release or therapeutic agents. This can also be used as a biosensor, medical devices, wound healing systems also can be used this type of polymers. The properties of these polymers is biocompatible, biodegradable, mechanical properties, surface properties also, whether it is hydrophilicity or hydrophobicity, and what is the charges present, whether it is negative charge or positive charge. You have to consider this while choosing these polymers for making the drug delivery devices. So types of drug delivery. Oral drug delivery is conventional drug delivery, what we are following earlier days, like tablets and capsules what we are taking, it comes under oral drug delivery. Injecting drug delivery systems, direct injections like syringe injections of drugs into the bloodstream is called injectable drug delivery. And transdermal drug delivery is an important, though we are not following much this transdermal. In foreign countries, they prefer this type of drug delivery system. People who are familiar with this drug delivery system will have good opportunities in foreign countries because they are preferring only this type of drug delivery devices for improved efficacy. These are examples are patches, ionotophoresis, and electroporation. And another type of drug delivery system is targeted drug delivery. This type of targets where actually it is required. Systems like liposomes, nanoparticles, monoclinical antibodies can be used in this targeted drug delivery. This detailed way I will explain in the next slides one by one. Inhalation drug delivery like nebulization and inhalers. Also, it is one type of the drug delivery system. And ocular drug delivery like eye drops and ointments. These and also implants like when we are using spectacles, it will not be convenient because some people they prefer these implants which can be used as a lenses, and this also can combine with medication also to improve the longer term efficiency of the eyesight and offering sustained treatment for condition like it is happening, it is doing both work. It is also giving medication to the system and it is also giving vision to the person. So drug delivery systems can be classified as follows. Liposomes, magnetic nanoparticles, niosomes, nanospheres, micelles, nanocapsules, nanoshells, solid lipid nanoparticles, dendrimers, grid gold nanoparticles. Nowadays, this gold nanoparticles has been taken into well shape because of its efficiency in drug delivery systems. So nowadays, lot of research is going on these gold nanoparticles. And drug limitations of conventional drug delivery is poor absorption from the site. If the medicine is unable to absorb the system, it cannot deliver the drug to the specific site. So because of this, we can use controlled drug delivery and define drug release, where it has to release and how much time it has to release. As per the latest advantage control release, we can modify. And target specify is a big problem in case of conventional, whereas in case of the controlled drug delivery system, this is not a problem, because we can use this because of control release purpose. Polymers are using because of their degradation rate is different for different polymers. So as per our requirement, we can choose the polymer combinations. So because of this, one more limitation because of this is premature excretion from the body. Before curing the system, it may be extracted because we are giving at that time that it is flowing through the stream, through the entire body it is going. So wherever it is, it has to act as a drug. It is not acting. Very minute quantity it is going to that particular point. So it may be not be efficient. So it premature excretion from the body may be possible because of due to conventional drug delivery system. Drug delivery protection from the chemical or enzymatic degradation hydrolysis, because of that polymers, it is mixed with the polymers, so it can be controlled. Whereas premature metabolism may take place in case of conventional, and improve convenience to the patient. So frequent dosage is not required. You can minimize the frequent, you can increase the frequency, and then it will improve the convenience of the patients also. So drug bioavailability is also poor in case of conventional drug delivery system, whereas improved drug bioavailability will be possible in case of recent advanced control drug delivery system. And this is poor patient to compliance. If you give, suppose if a doctor advised to take tablets in 3 times for 10 days. So patient may be forget, or he may wantedly skip this dosage. So this complaints will not be there in this patient complaints will be more in case of limitation. So you cannot follow these advices. Whereas in the case of recent control drug delivery system, better patient compliance is available because it is taking a long time. Suppose one tablet he has given, which is a sustained release system. So once in a day, if you take, it is comfort for the patient also, and he will follow the doctor's advice, and the effectiveness will be more compared to conventional drug delivery systems. Key benefits of drug delivery system is precision and targeting. Exactly where we want to deliver the drug, we will be delivering using this drug delivery system. And control release. How much fast we want to give, we can give. How much slow we want to give, or medium release if we want, we can control according to the selecting the polymers. Because polymer is a carrier for taking the drug into different parts of the body. So if you modify the structure of the carriers, then accordingly the drug will be released as per our requirement, and improve the drug efficiency and compliance, and offers also reducing the frequency of dosing. Because of this system, less side effects will be there, because it is directly going to the targeted site, and it is not going to the any other parts of the body, which is a good condition. So some of the conventional drug delivery system, if you give, it may damage the other healthy tissue system also. So this is the benefit of this drug delivery system. As I told, this patient compliance also will be good in case of drug delivery system. importance of control, improve the therapeutic efficacy and reduce the side effects, enhance the patient compliance, and this targeted delivery, minimize drug fluctuations. Suppose if normal tablet, if you give to the system patient, it will give fluctuations because of that nature of that polymer and the drug. In some of the parts of the body, acidity will be there. In some of the parts of the body, pH will be vary. Basicity will be there. Accordingly, it will not react. So wherever it is required, it is not releasing the, so fluctuations will be there in the release of the drug. So controlled polymer systems may provide this steady concentration of and a smoother or more consistent therapeutic effect. And it is also cost effectiveness. These are also though it is actually drug delivery systems are expensive, but our this cost can be compensated with a cost improving treatment outcomes. So it will be easily curable when you give the drug delivery system. So outcomes will be more compared to conventional drug delivery system, and reduce the hospitalization. Suppose if you want to go in hospitalization admissions also, it will be costly. So in that way, the cost of controlled drug delivery will be compensated. In that it will also reduce the need for more intensive medical interventions will be reduced. And the long-term options also can be chosen for this using controlled drug delivery system. Like chronic conditions, controlled drug delivery can be reduce the need for frequent hospitalization and visits and improve the quality of life for patient by providing a long-acting solutions for disease management. This actually I have removed, I have told already. So challenges with biodegradable polymers, maybe another problem is burst release. Sometimes the polymers may degrade very fast. So eventually the drug will be released immediately, and which may cause the side effects also. And these biodegradable cannot be control the degradation rates also. So in such cases like pH, temperature, and the presence of enzymes may be variability in the release profile. These are the having numerous advantages to improve like efficacy, improvement, safety, and convenience for pharmaceutical treatments. And drug delivery devices are designed to control the rate. It will also control the rate at which the drug has to release, the timing at the what time it has to release, and the location at where it has to release. And it also helps in optimizing the therapeutic effect outcomes also. It will be useful. So control and sustained release almost looks like similar, but these are slightly variation is there. Sustained release is a programmed rate of release dependent on external environment. This is comes under first order. The mechanism is under releases the profiles as per first order reaction. Whereas in control release system, it is determined that the rate of release will be predetermined and independent on external environment. It follows zero order mechanism. It will release the steady release drugs will be possible by using these systems. And drug concentration in the over period of time, it will be constant. It will help in maintaining the drug in a therapeutic window. This also reduces the fluctuation in the drug level that can lead to the side effects or inefficiency. Reduce dosing frequency. By controlling the release of the drug, DDS drug delivery system can extend the duration of action, allowing for less frequent dosing. Once daily dosing or even longer also possible are there some of the systems, drug delivery system. I hope some you might have seen SR in the tablet. Some of the doctors may prescribe the medicines with SR. What this SR is? Sustained release. This is the sustained release tablets. That means that frequency you can observe during your day activities whenever you are able to, whenever you have to take the tablets. The doctors should advise this type of sustained release systems. In that case, dosing will be less compared to other medications. It also improves the compliance by making treatment designs simpler and more convenient way. The advantage of other improved bioavailability will be there, enhanced because of bioavailability, it enhances the absorption of drug delivery system to the designed to improve the absorption of drugs. If the absorption of drugs is more, then it will release wherever it is have to absorb, there only it will be absorbed and releases closely. Whereas poor bioavailability drugs cannot be administered via traditional routes. So systems like liposomes and nanoparticles or prodrugs can enhance the solubility and absorption of poorly water soluble drugs. Biological barriers also can be overcome by this drug delivery system by engineering to biological, like suppose if the gastrointestinal tract is there, blood-brain is there, carrier or other skin, that limits the drug effectiveness. Suppose if you are giving some amount which is non-conventional tablets, it will flow throughout the body. So very limited quantity will go to the targeted site. So by using this control device system, we can overcome this type of biological barrier. Suppose if pH is there, suppose gastrointestinal tract will be having pH different acidity will be there. Stomach will be acidity and gastrointestinal will be higher pH. Like that, different parts of the body will be having different nature. So accordingly, it has to it will be overcoming by this type of control system. And targeted selective targeting, wherever we want to give the treatment, there only this drugs will be carried, and then there only it will release the drugs. Throughout the transportation, it will not release the drug. As per the because of the selected delivery systems has been tuned accordingly, like in case of tumors, inflammatory tissues or infected areas. You are giving tablets to the tumor. When it is not exactly going that very low concentration, it will be reach to that concerned site and will take long time to cure the disease. And it will minimize the exposure to healthy tissues also, which will reduce the side effects. Suppose unwanted tablets which are conventional methods, when you give that one, it may spoil healthy tissues also. So this side effects will not be there because of this drug delivery system, because it is pre-designed, where it has to design, how it has to design, what time it has to deliver the drug. Accordingly, we will be making these systems. So it will be not having any side effects. Improved efficacy. This drug delivery systems is directly to the site of action. Hence, efficacy will be more and therapeutic effect will be more in case of, it is important in case of cancer and autoimmune diseases and infections. So in case of infections, complete body will not be infected. Sometimes it may affect in a certain part of the body. So if your drug is giving through oral system, normal conventional methods, it may affect the other parts of the body. So, but actual infected body may not be cured, and it will take long time to cure. And minimize side effects also will be reduced by using this type of systems. This type of this is especially important for drugs with a narrow therapeutic window, which will be having a low immediate therapeutic window, means the therapeutic effect which is not be having any effect. Below that it will not be any effect, and above that it will be toxic. That is called therapeutic window, and I will explain with the picture in the next coming slides about this therapeutic window. I hope this will be important for the pharmaceutical people who are interested to go in the field. So accordingly, I have chosen this subject. Actually, I am not in this field currently, though I am not in this field because I have accepted to because of my friend. So I have given, I am presenting this with a hard effort. I have put this for making these things. So try to understand. Maybe some mistakes also may be there because I am not much familiar because of I have experience in my PhD. Based on that experience, and I have research area interest. So I have chosen this topic and I am presenting to you. And this is also minimized drug waste will be there in case of this drug delivery system, because it is used efficiently once by delivering the drug to specific location and for a longer periods, which will reduces the wastage and need for higher doses. Because when we are conventional doses, if you are using, it will not go to the complete system. So higher doses is required in a day. Because of this reason, drug delivery systems designed with a specific requirement may be helpful to reduce the dosage and frequency. Reduced pill burden with controlled release formulations, patient may no longer need to take multiple pills daily, which is not only simplifies the treatment but also reduces the number of medications consumed. Suppose if every medicine is having some side effects, in order to reduce those things, we are developing this drug delivery systems with controlled system and personalized medicines also. Suppose if a patient is suffering with some specific disease, accordingly this drug release profiles can be designed, and the dose that how much it has to be given and that where it has to be given, it will be customized by using this drug delivery system. And it is important for treating of specific diseases like cancer, where patients may require unique approaches based on their conditions and genetics. So improved treatment outcomes will come because of this drug delivery system, because due to overall success of therapy and reduce the side effects and enhance patient outcomes. And long-term therapeutic options also will be there in case of implants and depots. So some of the drug delivery systems, injections or implanted devices can provide long-term continuous drug release over weeks or months. In these cases, this long-term therapeutic devices can be fabricated, and which will be especially useful in chronic conditions like and preventing recurrence like cancer, hormone therapy, or in vaccination purpose. And it also reduce the frequent visits to hospitals. And it has enhanced the stability of the drug also will be enhanced because of this system. It is not directly exposed to the environmental conditions in the body. So it will be protecting from the degradation. Because drug will not be released in each and every place as per our design, it will release. So polymer will be protecting from the degradation of this drug. So like nanoparticles, liposomes, microparticles will protect these sensitive drugs from degradation of biofactors like light, oxygen, or acid presence in the stomach, and it will enhance the stability and shelf life of the drug also. And improved drug formulations can be made using this system by improving the solubility and stability of purely water. Suppose purely water soluble drugs also can be improved. It cannot be minimized. In minimum level, we can improve the formulations, because normally whichever drugs are soluble in water, then only it will be effectiveness will be more. So in order to have improved drug formulations, it will be helpful for non-soluble drugs also. And reduce the invasive procedures. Many DDS transdermal, like transdermal patches and inhalers and oral formulations can help in offering non-invasive alternatives to injectable or surgeries, and improves the patient comfort and reduces the risk of infection, pain, and complications associated with the invasive procedures. And reduce the healthcare cost also, it will be reduced. And reduce the need for frequent visits also will be helpful with this type of drug delivery systems. Improved management of chronic diseases like consistent release drug is required in some cases like chronic diabetes, hypertension, asthma, and other type of epilepsy. So study continuously drug is to be released for long term is critical for managing symptoms and preventing complications will be useful this type of drug delivery. And it also reduces the risk of disease fluctuations. Sometimes if you give the drug, the day it will be good. Again, so afternoon or by evening, if you are not taking the tablet, then it may fluctuate. Again, it will worse the health condition of the patient. So in order to avoid this thing, drug delivery can reduce the peaks and throws in the drug concentration by using this drug delivery system. Complications are disease flare-ups and improving overall disease management also can be improved. The drug delivery systems can provide significant advantage over traditional drug administration methods because they offer the potential for improved drug efficacy and targeted treatment and minimize the side effects and enhance patient compliance. It also helps in controlling the timing, location, and rate of drug release and allow the more precise and personal and efficient treatment, ultimately leading to better therapeutic outcomes and reduce the. Now we are going the drug delivery system, general drug delivery system we have explained. Now I will be explaining about the, by seeing this picture, we can understand this, the person is same. Here Rajinikanth is same, whereas his way looking style is different in both the cases. This is style is olden day style, and whereas this is recent style, that is new. Everything is the same person is, but way of this one's approaches is different. So in this case, this is just example I am giving here. This is a traditional, whereas this one is novel, like new approach. So definition of a new drug delivery system is a new approach to deliver the drug which addresses the limitations of the traditional drug delivery system. So novel drug delivery system refers to the approaches, formulations, and technologies and system for transporting the pharmaceutical compound into the body as needed to safely achieve its desired therapeutic effects. Novel drug delivery system is a system for delivering of drugs other than conventional drug delivery system. It is a combination of advanced technique and new dosage forms which are far better than conventional dosage system. The aim of this new novel drug delivery system is to provide a therapeutic amount of drug to the appropriate site in the body to accomplish promptly and then maintain the desired drug concentration throughout the treatment. For drugs to reach the targeted site with little or no side effects, it will be helpful by using this novel drug delivery system, and it minimize the drug degradation and loss to increase the bioavailability of the drug and fraction of the drug observed in the required site. So what actually new? Everything is new. What is now using this novel drug delivery system? Delivery system also is new, whereas formulation and dosage forms also new, and routes of drug administration also is new. Coming to this new delivery system, controlled delivery system, drug delivery systems comes under various, again, as per the requirement, it will be categorized as sustained release, zero order release, timed release, pulsatile release, and delayed release. This everything I will be explaining in my slides graphically. And whereas in the case of new formulations, dosage form, released erythrocytes, liposomes, nanoparticles through releasing of drugs, formulation of drugs using nanoparticles, nanospheres, nanosomes, nanosponges, microbubbles, and microencapsulation. Whereas the targeted delivery system in case of brain target, lungs target, this type of new delivery systems is available using this novel drug delivery system. And mucoadhesive drug delivery systems also can be used using this entities. So dosage forms also will be varied. Accusates, metered dose inhalers, implanted devices, buccal tablets, and buccal patches. So in some cases, the new routes, even we are administering the drug into the system by nail route. Means through nail we are injecting the drug to the system. We are directly not injecting to the body. Through nail it is, This is the novel technology for administering the drug into the system. And eye route also. Through eye route also, some of the drug systems is available to deliver the drug through eyes. This is called ocular drug delivery system. And this we can explain conventional drug delivery systems and novel drug. In novel drug delivery system, we have already studied this controlled drug delivery systems regarding. Now, targeted drug delivery systems, transdermal drug delivery systems, mucoadhesive drug delivery. In control, this is the control release system where the polymer will, along with drug, will be mixed together, and it will release, depending upon the mechanism, it releases the drug through the control polymer carrier. Suppose this is the target drug delivery. If we want to cure the tumor in the brain, we have to use the targeted drug delivery system will be using. Whereas through skin, if we want to give the medication, this we can call it as a transdermal drug delivery systems. And whereas the wherever wet absorption, by absorption, this one, because some of the polymers will be having adhesive properties, those type of polymers can be used in this type of drug delivery system. We will be putting this into the mucoadhesive wherever it is available. And the terminology related to the control system is defined as follows. Type 1 is targeted drug delivery, targeted release. If you see the tablets prescription, this one, cartoons, it will be written whether it is targeted release, whether it is modified release, whether it is controlled release, whether sustained release. Whenever, anytime you take the tablets, sometimes it may be prescribed this SR tablets or PR tablets, you can observe in the cartoons. The drug release from the dosage form will be to a particular organ. In targeted release systems, the drug will be released to a particular organ or tissue. Such type of drug delivery devices will be used in this case. Whereas in modified release, so it is modified from conventional release. So earlier days they used to normal tablets. So it used to give immediately to the bloodstream and it will release the drug. But it will not sustain much time. But this modified release will be to some extent time. So immediate release system is IR. This will release immediately to the, this is the called conventional release. Delayed release. Suppose whatever time we want, after some time, if we want to administer this drug, this type of system will be used. And next category is control release. In under control release, extended release, sustained release, prolonged release, time release, zero order release will be depending upon the at what time it has to deliver. This is extended release, extra time than that of the conventional system. It will take little time than to release the drug than the conventional system. It is more longer than the conventional. Sustained release is little longer, sir, compared to conventional. Whereas prolonged release will give much more longer time than conventional drug delivery system. Whereas time release based drug delivery system can be used in as per our predetermined at what time you have to give the drug to a particular specimen, or I will give these things examples in the next slides. Zero order control release system, independent factors and it depends only on time. Whatever the pH or concentrations it is there in the carriers, it will not depend upon that one. It depends only on time. So based on that time, it will release the continuously the drug. So this is the graph which we explain about these different types of drug release systems, control release, sustained release. Suppose if you give to see this number 1, so up to that some extent level it has increased and slowly it is decreasing. This is conventional release. This is what it will happen. This is, I told you that therapeutic, what is therapeutic window? This is the therapeutic window above which it will be toxicity actually, sorry, this is above which it is toxic. and below which there is will not be, if even if this concentration below this concentration, there will not be effect on that treatment. So this is called a therapeutic window. So coming to the second one, sustained release. Compared to first one, it has gone to slightly longer, and then it has reduced the concentration. This is also called as extended release. Compared to normal conventional release, it is extending the drug release time. And zero order, controlled or prolonged time. Number 3. See, if you see that number 3, it has increased and then continuously it is delivering the drug up to certain time. And a timed release. Number 4 is timed release. So it has been given, drug has been given. So up to certain level, it will not act anything. So after reaching to certain particular time, it will release. Why this is required, I will explain in the next slide. So then again, it will reduce its releasing rate. Pulsatile release. Number 5 is pulsatile. So like our heartbeat up and down, lab dub, so like that, it will up to reach, so releases the drug and stop releasing the drug. Again, release the drug and stop releasing that. Like this, it will be giving. This is pulsatile release. Whereas the 6th last one is delayed release. The number 6 is delayed. It will not give any drug will not be released to certain time, and it will be delayed release. Why it is required, I will be explaining in the next slide. So here, this is the patient with asthma patient is suffering from COPD. So he has to take the drug. Normally, patients who is suffering from asthma will be severe during late morning times, like 3 to 4 o'clock. It will be more effective will be there for them. They will suffer a lot. Because of that reason, whole day, if you take a tablet while sleeping, it will work immediately, and then when it is required, it is not releasing the drug. So it is called sustained delayed release. So he has to keep the alarm at what time he has to take the tablet. This is whatever is little inconvenient to the patient. In order to avoid these things, this concept has come delayed release. So here it will be the drug delivery system will be given such a way that it has to deliver the drug in a particular time. It will not be slowly releasing, but at a certain time, as per the determined fabrication of the drug delivery system, it will be releasing that, so that it will be comfort. It will be comfort for the patient also. I hope this concept, I think you might have understood. This old conventional drug delivery system has a lot of drawbacks, as I mentioned earlier. To overcome these things, new drug delivery systems are essential. So majority of the drug candidates suffering with a lot of loopholes like drawbacks or drawbacks. And these types of drawbacks can be overcome by NDDS. To optimize the biopharmaceutics and pharmacokinetics and pharmacodynamic properties of drug systems, and in order to maximize the drug utility, these type of drug delivery system can be useful in maximize the drug utility. Whereas in case of conventional drug delivery system, immediately it will be utilized. Wherever it is required, whenever it is required, that drug will not be available to cure that disease. So as per the disease demands, and it also reduces the side effects of drug candidate. It helps in curing the disease at earliest, because specific target we are giving, specific time we are giving wherever as per the requirement. So smallest possible time is required to cure the certain diseases. This also use the smallest quantity of drug as possible. Because in conventional system, if you take tablet for 3 times, here 1 time is sufficient. So that type of a facility is available with novel drug delivery systems. Some of the drugs will be having shorter half-life. That means it will not be having very half to reduce that 50%. It will not take much time. Like example, diclofenac sodium and the such type of drugs, sustained release formulation can be useful to use this type of drugs. So most of the drugs undergo first pass metabolism. Example, phenyl, only 30% reaches to systemic circulation. So most of the drugs undergoes first pass, means that time only minimum amount will be going to the circulation system, and 70% of the drug will be used for metabolizing before reaching the systematic circulation. So to avoid this type of things, buccal control release tablets are essential. So some diseases like asthma attacks at early mornings, as I explained earlier, patient cannot be able to take the medicine during the sleeping time. So in such conditions, this type of delayed release will be helpful. So patient will take medicine at night, but the drug will release exactly at the time of disease. Here, in this case, delayed drug delivery system, there is no need to wait till morning. You can take the medicine during the night itself, but the effect will be in the morning, because such a way this type of designs will be fabricated. And advantages of controlled drug delivery system: clinical, reduced in dose and dosing frequency, improve. These all things actually I have covered in the previous slides. Suppose engineers who are working in this pharmaceuticals, it is having people who are developed this type of systems will be having commercial values and industrial advantage also. Like a person who has developed this type of systems will get a name and fame for launching this type of technical products, and easy to get government subsidies also, and you can start the startups from the government, and patent benefits also will be used, and making money also you can make it. Please, so people who are working in this area will be having a lot of commercial benefits, and great market expansion will be there with the future in the future. And the selection of drug candidates for control, how to select the selection of drug also, it is important to make this controlled while using this controlled drug delivery system. No, whatever the exact whatever available drugs are not suitable to use in the controlled drug delivery system. So only drugs having following qualification can be selected for controlled drug delivery system. Biological or elimination half-life must be 2 to 6 hours can be used for controlled drug delivery system. Drugs with elimination of half-life of greater than 8 hours are generally suitable to such type of drug delivery system. So drug molecular weight should be around 150 for spherical shape molecules and must be 400 for chain-like compounds. Why this 400 is for chain-like and why it is 150 for spherical shape? Spherical shape, when it has to release means it will take time, because this shape is spherical in shape, because it will be inside also. Spherical means it will be some periphery, so it will take long time, so it has to reduce that molecular weight. So passing through this system, it will be difficult. So it has fixed as 150. And the molecules, because chain molecules, it will be loosely packed. So even 400 chain length having molecular weight of 400 would be suitable for this type of drug delivery system. So drug must have good solubility, at least 0.1% to 1%. Drugs which are having a solubility this less than this solubility may not be suitable for this type of controlled drug delivery system. And drug delivery bioavailability should be more than 60%. Drug steady state concentration must be low. Drug volume of distribution should be preferably less. And therapeutic windows should be broader as possible, so the formulator can design the formulations comfortably. Thank you for your patience. Please let me know if I could be able to answer your questions, please. Please, one or two questions or any doubts related to this.
Sir,
If
thank
I
you,
can
sir.
able to explain.
Sir, participant are there some questions, sir?
Yes, sir.
Regarding biocompatible polymers, what is the driving
sir.
force on a polymer to interact or compatible with living tissue?
What and sir?
Regarding biocompatible polymers, what is the driving force on polymers to interact or compatible with living tissue?
The polymer should be the functional group present in the polymer should be able to attack the fluids present in the functional group present in the fluids, whatever it is there. So it will be biocompatible. So it will not release any non-toxic byproducts. This maybe I think I'm not myself also not convinced with this answer, but
Okay, sir.
mostly this functional group will affect this mechanism, sir.
No. Most of the polymers used for the drug delivery is like a carbohydrates or else ethylene glycol. That is
Yes,
a
sir.
biodegradable and that can be easily passed out through a fecal matter.
Yes, sir.
It's
Yes, sir.
like a drug transfer, like a 𝐾𝐝𝐀 molecules are coming nowadays, like a linkers, and then head and tail. Like a drug is also like it will work as a head and tail, hydrophobic and hydrophilic. The polymer should be selected as a carbohydrate or else ethylene glycol should be designed in that way. Then only that polymer will be useful for drug delivery.
Okay, thank you, sir. Now it's vote of thanks by Dr. Erisam, sir.
On behalf of our institution, I take the pleasure to express our heartfelt thanks to Dr. Venkata Prasad for sharing valuable insights on polymer and its applications in modern medicine. We appreciate the time to share your knowledge with us. We are very grateful, sir. Thank you.
Thank you.
The second session will be start within 2 minutes. Sir,
So if you don't mind, I would like to leave because I have to go on some.
okay, sir.
Good presentation, Prasad.
Thank you, sir. Very
Thank
good explanation.
Thank you.
Prem sir.
He will connect you.
So my present is removed off, no sir? Delete, no?
Yes, I think it's removed.
నమస్తే సార్. బయలుదేరుతున్నా. ఇప్పుడు లెక్చర్ ఉంది ఒకటి, నేను మర్చిపోయాను ఆ రోజు మీకు చెప్పడం. 15 minutes 20 minutes లో సార్. వచ్చారా మా వాళ్ళు? ఓకే. Sorry, I thought my audio was off.
Okay, sir. So, good evening, sir. Prem, sir.
Yeah, good evening, everyone.
Can I start? Introduce, sir. Sir, please, your PPT will be present, sir.
Okay, shall I share my PPT?
Share, sir.
Just
Yes.
Is my screen visible to everyone?
Yes, sir. Okay, sir.
Yeah.
Sir,
Shall I continue?
wait, sir.
Yeah.
I,
Myself, Prem Kumar.
𝐾𝐲𝐶, Assistant Professor in Physics, with immense pleasure, welcome and introduce today's resource person, Dr. B. Prem Kumar, sir, who completed his doctorate in organic chemistry under the supervision of Dr. 𝐀𝐝𝐸𝐻𝐸 in Indian Institute of Chemical Technology, Hyderabad. He began his career as a postdoctoral doctoral research in Chang'an National University, Chang'an, South Korea, with various synthesis and catalytic applications of ruthenium, palladium, nanoparticles, and aluminum surface by chemical co-precipitation and a consign synthesis of 4E10Z and tetradeca for 10-dinyl acetate, sir. Sir also is specialized in research of organic inorganic hybrid materials, its applications. Also novel diethanol as capping ligands for cadmium, selenium, quantum dots, optical properties, and solar cell applications. Sir worked as junior research scientist in Piramal Healthcare Limited. Goragan worked as senior principal scientist in Sai Life Sciences Limited, Hyderabad. Later, Sir worked as head of chemistry in Sambi Sciences Limited, Hyderabad. A profound efficiency in handling reaction, expertise in analyzing data needed for structure and illustration, designing innovation strategies in asymmetric synthesis, and so on. Sir has contributed a lot of several outstanding publications on notable synthesis of both organic and inorganic with remarkable application that have an recognition and advancement of knowledge. Sir has received prestigious patent that demonstrate exceptional creativity. He has presented posters in the 107th KCS meeting at South Korea, 12th CRSI National Symposium in Chemistry, the 4th CRSI RSC at IIT Hyderabad. Sir has participated in several national and international conferences held at IIT Hyderabad, Delhi, and so on. He is well known for independent research, very easygoing, and an amicable person. It is a privilege to have you, sir, with us as the resource persons. We welcome you, sir, and request you to take over the session, sir.
Yeah, thank you welcoming me.
Thank you, sir.
Yeah, briefly going into my research means my background. I started my career during my PhD in total synthesis. It's a multi-step total synthesis containing of bioactive natural product synthesis, which is of 20 to multiple steps, 20 steps, 20 steps, 30 steps, including many reactions. And after that, in South Korea, it's a different area with material science, surfactants, and other areas, pheromone synthesis. And after that, pharma industries working in API and API intermediates in new drug innovations. And with the experience of South Korea with the material science, I got an opportunity to work in Tata Steel. There, I love to work in different materials for iron and ore coal beneficiation by designing a surfactants with different surfactants, which is sustainable surfactants and biosurfactants. They achieved many patents, around 10 patents and few publications. Then afterwards, they called me back to Hyderabad to work in pharma industry, Life Sciences, to work in a new area like nucleosides, DNA molecules and RNA molecules. At that time, this COVID is COVID is there. At that time, I worked for remdesivir and then different molnupiravir and other molecules. It's a drug delivery like Galnac molecules and other. I have given suggestions to Gilead also by giving the few molecules, and it helps a lot. And we have given molecules to Bharat Biotech. And this is my experience. And going into the our few topics of nanoparticles. This how the nanoparticles are important, how the nanoparticles are synthesized, and how it is applicable. And here, few of my publications and areas I worked is iron nanoparticles and catalytic activity in click chemistry, cycloaddition, and magnetic and other work is a bimetallic of ruthenium and iron particles. And one more area is ruthenium nanoparticles and applicable in click chemistry, and palladium nanoparticles, which is prepared from co-precipitation. And lastly is luminescent materials and their applications. And going into the definition of nanoparticles, nanoparticle is a tiny material consists of 100 nanometers in diameter, and it is invisible to naked eye. And what are the properties with the existing of, if you take the palladium palladium carbon, means palladium carbon, many of the people will use for reduction. And why it is important to have a nanoparticles? Nanoparticle is having a different properties, chemical properties and the physical properties. When we compared with the bulky material like other particles, other materials, and its surface area is more, and it is capable to have a high efficiency reactivity, and the nanoparticles having different shapes and polymorphs. And the usage of these nanoparticles are used in catalyst and drug delivery and gene therapy, and it is used in biodetecting in the organisms by injecting in a mice or else where targeted disease, and biosensing and the gas sensing and carbon dioxide capturing. So nanoparticles are used in many variety of applications. And going into the iron nanoparticles are synthesized by following this iron chloride reacted with ammonium hydroxide. This ferric chloride is dissolved in water, and then after that it's added into the homogeneous solution of ammonium hydroxide. When it is adding into the ammonium hydroxide, iron oxide is getting precipitated by the release of ammonia and water, and this is creating a nanoparticle. When we are characterizing is it a nanoparticle or not, and we submitted for TEM, and it is showing the tiny particle, and it is characterized by FTR also, it is showing, and submitted XRD, and it is giving good results, and the magnetic property is also good. And after having these nanoparticles, and we applied for different reactions like having the azide and alkyne, it is linked with the cycloaddition to have this reaction. We call it as a click reaction. and it's a cycloaddition reaction, and we have done with different examples. And we have done means after reaction we filtered and we recovered that material and reused for almost 15 times. In this way, this nanoparticle is useful, and we can recover and we can reuse so many up to cycles will be there, up to 15 times we used, and this activity is also good. And this is a possible mechanism. This iron nanoparticle is chelated with the alkyne with the terminal acidic proton to have a Fe, and then afterwards is bidentate, and it is chelated with the azide, and it is cyclized to have a triazole moiety. Then we call it as a click reaction, and after having the triazole moiety, it will cleave this iron, and again it will participate in one more molecule. This is a possible mechanism for having the cycloaddition reaction. This one more bimetallic complex of nanoparticles, ruthenium and iron. Why we are going into the bimetallic complex? If you take ruthenium is expensive and iron is a cheap, the function of this ruthenium will be the same, and iron will initiate the reaction. That's why we selected that ruthenium and iron bimetallic complex. This bimetallic ruthenium and iron nanoparticles are prepared by using ruthenium chloride and iron nitrate by mixing with the same surfactant, and after precipitating, filtering, and calcination at under 300 degrees Celsius to have a ruthenium iron nanoparticles. If you see the below equation, this iron nitrate plus ruthenium chloride and sodium carbonate, and it is giving the ruthenium and iron nanoparticles. And after having this iron and ruthenium nanoparticles, we characterized with XRD, FTIR, and the TEM and SEM with the EDX elemental analysis, and it is showing this ruthenium and iron nanoparticles. And this is having the huge application like methylene blue absorption and desorption at different sunlight, sunlight and UV light. If you see the graphs, and we can easily detect that absorption is low at UV, and at sunlight it is high. And the preparation of these ruthenium nanoparticles, and we prepared the same like ruthenium chloride we have taken and precipitated in the CTAP, polyethylene glycol, and after having this precipitation, and we filtered and centrifuged and calcinated, washed with acetone, calcinated at 500 degrees Celsius, and obtained a black precipitation with ruthenium nanoparticles. And we characterize that ruthenium nanoparticles with the SEM, TEM, and XRPD, and it is showing the nanoparticle size and elemental analysis. And the same application we have done with the click reaction azide and alkyne. And we have done with a different tri-substituted alkyne, and then with the dodecyl azide, and benzyl azide with a different triazine alkyne, trialkyne, and it is giving a good results. And this application can be used to link with a different polymers or else a different drug to have a linkage, and this can be used for self-healing material also. And the mechanism will follow the same as iron, and chelated with alkyne acidic proton with the ruthenium, and then this ruthenium will chelated with the azide, and then cyclized to have a triazole moiety, and then after removal of this ruthenium, and again it will come to ruthenium 0 and again reparticipated. And the cycle of this ruthenium is around 20 cycles, and for 20 cycles we filtered and we reuse that ruthenium nanoparticles repeatedly, and it is very useful and sustainable ruthenium nanoparticles. And the palladium nanoparticles on aluminum surface. How this palladium nanoparticles aluminum surface is synthesized? By taking this palladium chloride and aluminum nitrate and having this homogeneous solution, and then mixed in the ammonium hydroxide to have a precipitation, and centrifuged and washed with acetone and dried at 500 degrees Celsius and calcinated to have a palladium aluminum oxide nanoparticles. And if you we have characterized with the SEM-TEM and all this data EDX and applied in a different Suzuki reactions and EMR coupling reactions. These reactions are very useful for drug synthesis. And if you take the trimethoxy benzene silane and iodobenzene, and we can couple with the dibenzene compound to have with the yield very good yields. And we have done with the different examples of bromo compound, iodo compound, and with the different triethoxysilane compounds. And this is a coupling of iodo and triethoxysilane is called as a Yama coupling reaction. And if you go for Suzuki reaction is a coupling of boronic acid with alkyl halide. And we use this palladium nanoparticles on aluminum surface. It's giving a good results, and we used for reduction of alkane and alkene to alkanes, and it is giving good results. And the cycle reuse and recycled and reused up to 15 cycles. And these different organometallic applications. This metallic application we used for metal organic frameworks and covalent organic frameworks. And how this Korea and Japanese people are preparing, most of these are used for electronics materials, and then devices, and the water filters, and the polymers. And we can prepare with a different having the same like a defined structure, and we can pass and we can capture the heavy metals. If you see the metal organic frameworks, the compound clusters with the metal and then organic linkages. And this metal is linked with like heteroatoms or else olefins, and to have a defined structures. And if you take the covalent organic framework, and this amine will react with the aldehyde to have a oxone, and it will form a different polymer structure. And these applications are used in a different application like filtrations, capturing of heavy metals, and purification of oils, and different application drug delivery. Many applications these MOFs and COFs are used. And a few of the examples we have done with the cadmium selenium quantum dots. We prepared the cadmium selenium by using the cadmium chloride and selenium oxide, and same calcinated property, and having these quantum dots, and the quantum dots are chelated with the dithiols, and dithiols are prepared by using this triazine. We call it as a cyanuric chloride, and cyanuric chloride treated with the sodium bicarbonate by using the two equivalents of sodium thiol, and with the different amine compounds. And in one part, we prepared this dithiol compound, and having the head of a different, like this is a carbazole, and this is a dibenzyl compound, and morphine, and this is a pyrrolidine. And after having this different dithiol compounds, it is chelated with the cadmium selenium quantum dots. And this each material is having a different color properties, and these materials are having different chromophore and different UV absorptions. And each material is used for a different application, like we used for solar cell energy conversion. DSSR, disenchant solar cell preparation. We prepared the solar cell by having this titanium layer, And then we have poured this electrolyte, and after having this chromophore, and we got this some energy of light, and this may be useful for dye-sensitized solar cells. And one of the organic inorganic hybrid materials which is having the selenium and organic compounds, and this looks very big moieties, a polyhedral 𝐀𝐝𝐶, but it is very simple to synthesize by having this tetraethoxysilane, and with a control addition of uh, water, and we can get this trial moiety opened at one side. This trial moiety can be linked with one more chain tetraethoxysilane, and then afterwards we can chelate with any heteroatom or else acid chloride, acid or else, and we can make any bond. And this having these heteroatoms, it can be chelated with europium or else any metal, and it will give some color absorption, means color at some UV range. And this material can be used for luminescence materials, which can be like a bioimaging or else sensor materials or tagging materials with different applications. And the same luminescent material we prepared by having the same tetraethoxysilane, and one side open with a trial moiety. It's a one-step process to have the chelated with a europium. And if you see the absence of UV, and in presence of UV, it is showing the red light when it chelated with a europium. And this is a mesoporous material. The mesoporous materials are prepared with a different polymer. If you take the monomer and having this any surfactant, if you dissolve the surfactant by agitating into a small micelle with a different surfactants, if you take a different different surfactant is having a different micelle size, and around that micelle size, this is a micelle. If you take it as a, this is a micelle. And around this missile, and if you make it polymer, and all this polymer around this micelle, and after removing this micelle by dissolving in a different solvent, extraction in ethanol or else heating, and it will create one hole, a small hole. And these holes can be used for different applications like fuel cells or else carbon dioxide sequester, then water filtrations, and then some stereochemistry materials also we can filter, and one isomer will stay upside, and one isomer will pass it. That technology also now people are developing, and this is a useful technique for different applications. Pollution control also we can do by capturing different heavy metals. Yeah, this is a small presentation from my side. If you are having any questions, I'm ready to answer.
Sir, participant asking one question, sir.
Yeah.
Request you to organize the procedure of ruthenium and iron nanoparticles, sir.
Yes, sir. I want to ask, please give me the procedure of ruthenium nanoparticle and iron nanoparticle.
Yeah, we have given the procedure also here.
Yes, sir. So can you share this slide with us?
Yeah, sure, definitely.
So
I
actually,
think already Dr. Swami is having these slides, and you can have it, and you can prepare, and you can apply it for different organic reactions. And
yes, sir.
the people means this engineering people can prepare a different bimetallic. If you take rhodium or else iridium metals, these metals are very useful in the industries. The people are wasting lot of metals, precious metals. To have this one, and people are preparing this bimetallics with iron. Iron is acting as a enhancing metal, and by having this ruthenium or rhodium.
Okay, yes, sir. Already we have prepared here iridium nanoparticle. Actually, I'm working in East Point College in Bangalore. So their facility is good. So,
Okay.
but now we want to prepare here iron nanoparticle, ruthenium, and rhodium. So I can get help for this for your slides only.
Yeah,
So
sure,
if you,
definitely.
okay, sir. So I should contact to Swami, sir.
Yeah, Swami is. I shared this PPT with Swami, and
Okay.
he will share with you people. Or else,
Okay.
if you send a mail, or else Swami will coordinate, and whatever the information you want, I can give. And I will share my email ID with Swami, and he will share with everyone.
Okay.
Sharing
Thank you so
WhatsApp
much,
group
sir.
also, madam. PPTs.
Thank you, sir. Thank you, sir. Thank you, sir.
Okay, thank you.
Yeah. Any other questions, please?
So only participant asking only one question. That question, sir.
Yeah, this take by if you see the equation, iron nitrate and ruthenium chloride and bicarbonate means this is a bicarbonate for precipitation, and after having these two metals are dissolved in a water, and this is also in water, and this iron ruthenium and iron is insoluble, and if you centrifuge it, and we will get the precipitation, and if you calcinate it, and then you will get the nanoparticles.
Thank you, sir. Yes, thank you.
Yeah, we can play with a different different metals to have a bimetallic complex, and we can apply with a different applications. Nowadays, a flow reactor is there. Directly, we can prepare one cell or else a column, and we can pass through the reactants, and we can get the directly the product. Yeah. Anything else, please?
Nothing, sir. Now it's vote of thanks, sir.
Yeah. Thank you.
So now it's vote of thanks. So on behalf of our institution, we extend our special thanks to Dr. Prem Kumar sir for enlightening us with a remarkable presentation that has enhanced our understanding of this cutting edge field. Thank you, sir.
Thank you.
Participant, I will post that feedback form in chat box, WhatsApp group.
Okay, sir. We will send it.
Thank you, sir.
Thank you, sir.
Thank you, everyone.
Thank you, sir. Thank you, sir.
Thank you.
Thank you, sir. Participants, sir, all of you.
Thank you, sir.
Shall I leave this, sir?
Please, sir.